So good afternoon. My name is Faith Dukes. I am at Lawrence Berkeley National Laboratory, and I manage our K-12 STEM education programs here in Berkeley. And today you are joining us for our sourdough experiment. So we're going to learn how to make our own sourdough starter, as well as learn about some of the science behind it. So you'll see the list of names here: Dr. Jennifer Johnson, Mariah Parker, Josh McCauley, Robert Riley, and Leo Baumgart are all going to join us over this next hour and a half and help us understand a little bit more about yeast, bacteria, and sourdough. So just some quick reminders as everybody is joining us. The first thing is that we want to make sure, if you haven't already, there is a worksheet that we actually created for this that you can go back and reference later or reference right now. You can get that from k12education.lbl.gov, and it's on our home resources page. Under that link, you can find our live science series. We've highlighted it here on the slide, and you can check out the rest of the science series that we have coming up for the rest of May, but also download any important information. Yes, we are recording this, so this will be updated later and put and edited on our website, as well as we invite you to ask questions. The whole point of doing this live is to allow you to ask questions in real time. There is a question and answer box. We will be monitoring it over the course of this time period, and we will be answering it as we go along. If you would like to follow us on social media at Berkeley Lab or LBNL Next, that's our K-12 office, please make sure you do. And for those of you who do start a sourdough starter, use the hashtag LBNL We Rise Together. So just a little bit of rules of engagement, as well as some reminders and information. For the beginning of this, we'll introduce the topic, then we'll do our experiment, get a little bit more science, and then go outside with Leo, who is turning his garden or has his garden and also a laboratory at the same time. So just to give a little bit of background, we are Lawrence Berkeley National Laboratory, one of 17 Department of Energy national laboratories in the country, and that's us all the way on the West Coast. Here we do fundamental research, and we have been doing that work since 1931. If you're interested in learning more about some of the laboratories, you can actually visit symmetrymagazine.org, which has an this interactive map actually, and allows you to learn more about the laboratories. Here's a look at our view, one of the best views from the lab at the top of Berkeley, where we have 200 acres and over 100 buildings that do innovative research. And that's usually our look from the lab with our turkeys, who are taking care of the site while we're not there and we're working from home, because today we are joining you from all of our homes as we talk about sourdough and sourdough starters, and a little bit of the science behind yeast and the work that some of our scientists do studying yeast. So the first person that I'm going to introduce to you is Dr. Robert Riley, who is a data scientist at the Joint Genome Institute, and he's going to introduce the rest of our topic. So I'll transition over to you, Robert.
Thanks, Faith. Well, hey there, I'm Robert Riley. I work at the Joint Genome Institute, also called JGI, where we do all kinds of science that is related to the genomes of a bunch of different kinds of organisms, plants, fungi, as well as metagenomes, microbes. And next slide, please. So I want to start with some vocabulary that we'll be using throughout this talk and throughout this, this experiment we're doing. So I just want to tell you what these things are: fermentation, microorganism, blah, blah, blah. Now, fermentation is a biological process by which organisms convert sugar and things like the starch in your flour into stuff like alcohol, acids, carbon dioxide, etc. And the next thing here, and so what is a microorganism? It's a unicellular organism that you usually need a microscope to see, and it includes bacteria and yeasts that we'll be using in our sourdough experiment, but it also includes things like archaea, protists, and algae. And so what is the genome? I mean, we have it in the name of our institute. Well, the genome is the genetic material of organisms made of DNA for almost all organisms except for RNA viruses. But for these purposes, it's just the genetic material of organisms. It's the DNA that's in an organism. Bacteria, those are single-celled organisms. So organisms with only one cell, and they have a cell wall, but they don't have any organelles, which are little compartments within the cells, such as mitochondria, which generate energy, or nucleus. And they have, in general, a single circular chromosome that their DNA is makes up. Next slide, please. So yeast, well, rather than just show you another picture of unicellular organisms, they are also unicellular organisms, except they are fungi, not bacteria. They're similar in some ways. They have a cell wall, but they do have organelles and nucleus, and they have multiple linear chromosomes consisting of their DNA. Next, please. Waiting for that next animation to come up. So microbiome, what is that? It's the collection of bacteria, yeasts, and other microorganisms, all of them together, that are present in an environment, including your hands. Something to think about is that there's lots of microscopic things living on your hands right now. And next is acid. Acid is just a chemical, including those that give some foods a sour taste, and it's typically lower pH, less than 7. And that's relevant to our sourdough because we're producing acids to make our sourdough bread sour. And so what is sourdough starter? Well, it's a mixture of flour and water in which the naturally present microorganisms or microbiome have been allowed to ferment for a while, producing these acids that give sourdough their taste. And this is an ingredient, or a starting point for sourdough bread. Of course, you need the dough later on when you go to make the bread. But anyway, this is the goal here. This is what we're making in this experiment, and that's what sourdough starter is. So a little more on just unicellular versus multicellular organisms so that it isn't confusing. So unicellular organisms, like I said, they're just an organism with a single cell. They can be bacteria or yeasts. They can also be archaea and algae. As opposed to a multicellular organism, which is an organism where an individual is made up of many cells, trillions often, in the case of a cat or a goldfish or a tree. And next slide, please. Now, a little bit more on bacteria. Like I said, they're unicellular organisms with cell walls, no organelles, no nucleus, linear chromosomes. They all look pretty much the same under a microscope. They're just these little rods or spheres or something like that. But although they look all quite similar to us, they're actually quite diverse, and there's a lot of them, of different ones with different ecological niches they occupy and biotechnological or physiological properties. So there's some 500,000 different species of bacteria in the NCBI database, and as we're constantly discovering new bacteria and other microorganisms through metagenome research that we're doing and that's being done around the world, there's certainly way many more bacteria out there that we don't know about. Now, as far as just in your own, microbiome, there are, it turns out, trillions of bacterial organisms on and in your body, mostly your gut, but also your skin and other places, outnumbering your own cells 10 to 1. So in terms of number of cells, one way of thinking about it is that you're more bacteria than you are human, at least in terms of, by that measure. not really true, but it's just to let you know that bacteria are everywhere, including right there with you. And then, so like I said, the role of bacteria in sourdough is to break down the starches and things into yeast. So what are yeasts? Like I said, they're unicellular one-cell fungi. They also look very kind of boring and nondescript under a microscope, but they're really quite cool, and I'll get to that later. And this is just opposed to other kind of fungi, the filamentous multicellular fungi, which under a microscope, they look different. They form these filaments. They can grow into these macroscopic structures, which you've seen before, like mushrooms, including the humongous fungus, which occupies 3.5 square miles up in Oregon. It is thought to be the largest individual organism on Earth. So again, there's a lot of different kinds of yeast. 6,000 species of them have been described in the NCBI database, and we're discovering new ones all the time. Like bacteria, the yeasts are everywhere, including your microbiome, although they make up a lesser proportion of your microbiome than the bacteria do. So why is the sourdough bread sour, and how does that really happen? And what do the bacteria and yeast do that allows that to happen? And this is how it works. So here's a yeast. It's hungry. Yeast love to eat sugar. So the yeast finds some sugar, and then it eats the sugar. And then it's very happy and full, and its stomach starts growling. Well, it doesn't have a stomach, and then it burps CO2, or it exudes CO2 gas that is typically a product of some kinds of fermentation, and then ethanol, perhaps, although it can also result in the case of sourdough, lactic acid or acetic acid, etc., other organic acids that give things a sour taste. So although this process is depicted with respect to yeast, the bacteria also do a similar process, just using kind of different biochemical pathways in our sourdough starter. So what's the relation between your own microbiome and the microbiome of your sourdough starter? Well, here's a figure from a study that I read on the microbiome of sourdough. And on the left, we have a picture showing the yeast present on bakers' hands, the microbiome of bakers' hands, and the individual bakers are labeled A through R in the figure. And it shows you the diversity of the microbiome of their yeast on their hands, and then in the sourdough starter produced by those individual bakers. And it's interesting to see that although the hand microbiome composition of fungi is quite diverse on the individual bakers, it mostly turns into the same profile of organisms when the starter is allowed to sit for a while. So that's kind of neat. Despite bakers' microbiome differences, the sourdough communities mostly become similar. And just on the right is the same thing, but with the bacteria shown instead of the fungi. So it was also found in this study that the sourdough microbial communities also influence the microbiomes of the bakers. So if after some time, the baker's hands microbiomes was measured again, it would tend to resemble the sourdough starter that they've been working with. So this illustrates the idea that there is always an interaction between your microbiome and that of the environment. So it's a good reminder that there's always things living on your hands, and it is a good reminder to wash your hands, especially in the times that we're living in. So this is how you wash your hands. You go for 20 seconds with warm water and soap. You get those thumbs, you get both sides, and then you're good. So it's a good reminder to us all. And next up, the experiment.
So thanks, Robert, for giving us that quick introduction and a little bit of science behind things. We're now going to turn it over to Mariah Parker, who's a postdoctoral scholar here at Lawrence Berkeley National Laboratory, studying very reactive materials, and Joshua McCauley, who is a research scientist over at in Emeryville at our J-Bay site, who's studying also genomics. and who is a kind of master baker and sourdough starter and can give us a lot of information over the sourdough breads that he's made. So I will turn it over to you all and stop sharing my screen so that we can see them both.
Alrighty, hi everybody, my name is Josh, and I've been making sourdough for about 2 years. I started doing it when I was doing science in another lab that had nothing to do with microorganisms, but I still wanted to experiment with them. So I started making sourdough at home to work with the microorganisms, and I've been doing it ever since.
Hi everyone, my name is Mariah Parker, and I have been baking for about a month now. This shelter-in-place time has really given me a really unique opportunity to really explore new things and like dive into the chemistry and the science that's happening here. So today what we're going to be doing is we're going to be making our own sourdough starter. So really we're making an environment where this bacteria and yeast can flourish to make our sourdough bread. So first what I'm going to do is I'm going to go over everything that we need today and then give you guys a couple minutes to gather anything you may need before we really start. So first things first, if you have an apron, go ahead and put that on. Josh has a really cool one on right now, and that would be considered our safety equipment for today. You're also going to need some sort of container. This is an old salsa jar. It can really be anything. It can be an old yogurt container. It doesn't have to be see-through, and it definitely doesn't have to be glass. You just want to have something that has some sort of closable lid on it. You're also going to need flour and water, preferably at room temperature. That just tends to work a little bit better. Also some tape and a marker, as well as spoons, measuring cups and measuring spoons, or a kitchen scale. So while you guys give you guys a couple minutes to do that, I have a question for you, Josh, just to review some things before we get into the science. What is yeast?
So from what we learned from Robert, yeast are single-celled fungal microorganisms. And as humans, we use them to make a number of things, such as bread, beer, and a lot of medicines. Mariah, my question to you is, what are we doing today?
So today what we're going to be doing is we're going to be starting our sourdough starter. So we're going to be making an environment for those particular bacteria and yeast to flourish so we can eventually make some sourdough bread. So I actually made some bread from a starter that I already have yesterday to show you guys, and I have to say it is really, really yummy. So get excited about our breads today. Another big thing is yeast are really, really important. So what would happen if we didn't have any yeast? What would happen if they all went away?
So that's a really good question. So a couple things come to mind. First thing is we probably wouldn't have bread because that's what we mostly use to rise it. And we wouldn't have a lot of the beer and medicines and other foods that we make yeast with. But it's also really important to note is that since yeast is a very important model organism in a lot of biology, if we didn't have it, we wouldn't have learned a lot about human genetics that we learned from using yeast. But thankfully, yeast are everywhere, and today we're going to start growing them in our own kitchens.
Perfect.
So,
so
guys,
Josh, what is going to be our first? Oh, sorry,
before
Jennifer,
we get started, we have a question from the audience that I wanted, if you could address. So the question was asked during Robert's talk, but I thought maybe you guys could answer it. What is the difference between regular bread and sourdough?
I guess I could take that one. So when you buy bread at the store, it's typically raised with either commercial yeast or like baking soda or baking powder. So when yeast is used to make those breads, it's a pure culture of yeast. And so that's why you don't really get sourness in a lot of breads that you buy. Whereas sourdough starters, it's a population of both yeast and bacteria. So that's why it is sour, and you have to be a little more careful with it in the way that you make your bread because it's not as robust as the pure cultures of yeast you can buy at the store.
Perfect. So let's get started. Josh, you're going to walk us through how to make our starter. So what's step number one?
All right, so first things first is you're going to want to measure 60 grams of flour, which is about a half cup unpacked, so don't pack it very tight. So let's get that. So you're going to want to have your half cup of flour, and you're going to put it in your container. Then now, similarly, we're going to put 60 grams of water in the same container. So this is about a quarter cup of water. And you want to make sure that it's cool or room temperature, because if it's too hot, you're going to kill those yeast and bacteria.
Hey, Josh, do you sift your flour?
I do not. But that's a good question, and it may or may not be a good idea, depending on if you're starting with whole wheat, rye, or any other whole grain flour. Here I'm using all-purpose, so it's already sifted. Okay, so once you have your quarter cup of water, you're going to mix it with your flour, and we're going to stir it until smooth. So at this point, it's very important that you start making some observations about your starter. So first thing is you want to pay attention to how it looks. So it's important to note here is that if you use cups or tablespoons to measure out your ingredients, you may see a little variance in how your mixture is going to look. That's because cups and tablespoons aren't the most uniform things ever. So what you want to look for is you want to get a consistency that's kind of like a soupy oatmeal, about the consistency of molasses, if that makes sense too. So mine is actually a little dry, so I'm going to add just a little bit of water.
Josh, what do you mean by if it's a little dry? Does that mean that you can still see some flour in there, or is there a type of consistency that you're looking for specifically?
Yeah, so it's a certain consistency you want. So if you get your spoon, let me get it to a good point with mine. Let's see, I need to be a little taller. So yeah, what you want is you want your starter to be about this consistency. If it's too wet, you're going to want to add flour 1 tablespoon at a time until you get this consistency. And same thing if it's a little too wet, like if it's essentially water, you want to add your flour about 1 tablespoon at a time. So what's really important is that you get this right ratio of about 60 grams of flour to 60 grams of water. This is the ideal ratio for your starter. So it's okay if when you're measuring, you're not the most accurate in the world, as long as you can adjust and get this consistency that we're looking for. So
I'm also just going to go ahead and show mine. So that's also what mine looks like. It's that really thick oatmeal-like consistency. It's going to look kind of boring and by boring, I mean it's not going to be doing that much for the first couple days. It's probably going to look like this, maybe even get a little watery. And then before we move into the next steps, we're definitely going to talk about what's going to happen. But I do just want to say, give that shout out now. And then what we want to do.
Go ahead and, sorry, can you show your mixture again just for a little bit longer?
Absolutely.
Thank you.
So this is what mine looks like, where it can have like that little bit of a flow. You don't see any dry flour in there anymore. It is all incorporated into this mixture. And then it's in its little container.
Mariah, before we move forward, I want to make a point that it's really important right now that you make some observations about your starter. So, at least for me, smells just like the flour I put in it. Nothing stinky going on there. It's about as consistent as we were talking about, and it's not very tall. It's important to note these things because you're going to see a lot of changes in the next couple of days and weeks, but we'll get to that in a bit.
So the next thing that we're going to want to do, just to make sure that our starter gets really happy, is you want to cover your starter because you don't want anything else getting into our starter. a good thing to note is that you don't want to make it too tight, because like Robert talked about before, the yeast and bacteria are going to be releasing those gases. And if your container is too tight, those gases won't have anywhere to go, and your container might break, which can be kind of dangerous. So you do want to make sure that it's closed, but you don't want to yank it too tight. We're also going to label our starter. Like, we're all scientists here, and we all want to make sure that we're keeping notes and we're making sure that everyone knows what's in our jar. So one, they don't throw it away, because we don't want that. And two, they don't eat it, right? This could look appetizing to someone, and we don't want that. So what we're going to do is we're just going to take some tape and we're going to put it on our jar, and we're just going to label it that this is a sourdough starter, and we're starting it on May 8th. I almost said May 7th. So I'm just going to go ahead and label that here, that it's a starter, and that today's date is 5/8/2020. And you also want to take a second piece. I like to use 2 pieces of tape. You can use 1 piece of tape if that calls your name. And I like to make a tally mark of what day it is. So here's my final label. So you have your starter, today's date, and then today is day 1. And you're going to make a tally mark for every day that you're feeding your starter. We will go into how to feed that in the next couple days in a minute, but you are going to make a tally mark just to keep track of how long you've had your starter and how old it is. Cool. So then once you have that, that's going to be ready. You're just going to put it somewhere that's not too hot and it's not too cold. So now that we have that piece out of the way, I'm going to ask you, Josh, what are we going to see over the next couple days with our starter?
So you're probably going to see a lot of things. First, you can kind of go over smells. So starter is going to change a lot in the way that it smells. Everybody has their own opinion on how they think it smells, but it could be anywhere from smelly to fruity. Depends on what happens with your starter and how you feel about it. So in the next couple of days, that's probably going to change a lot. Might get really, really stinky, but then eventually it may turn into like a fruity smell. Some people say it's like a fruity pickle, or
I think they smell like really dirty socks, and it's going to smell really stinky, and you're definitely going to make some faces when you smell it. But I want to definitely underline the fact that that is totally okay. What's happening is that the yeast and the bacteria that we want to be growing is starting to fight the other types of bacteria and yeast that are present in our flour and water, and they're fighting to make sure that they're the ones that stay. And that's why it smells really, really bad. What is another type of observation that we're going to see over the next couple of days?
So there's a lot of visual things that you can see with your starter. So you're going to start to see a lot of bubbles. So you'll see that your starter gets very bubbly. And because of that, it's probably going to change a lot in height. So it may get taller than it originally was when you're making your starter now. And that's actually great. So the goal for our starter is to have it double in its height in about 12 hours. At that point, that means that it's vigorous enough that we could start making bread. So for example, here is a starter I made this month. It is quite bubbly, and it is very much double the height that it was when I fed it, at least this morning. So it's doing quite well, and I can make bread with it.
Yep, I do just want to say, and just to underline the point, so this is the starter that we just made. It's this height, and this is where your starter is going to be every time you feed it. And then what's going to happen is it's going to grow up. So this starter is actually 28 days old, and you can see that in comparison, that does grow quite a bit. This is actually probably over doubling in size, and this is a really, really happy starter that we've made, and I'm actually going to go ahead and feed that now with you guys today. But it could take some time before you guys see all of these bubbles. Sometimes, depending on our starter, it could take as little as like 5 or 6 days to see that many bubbles, and it could also be like this one took about 2 full weeks before I saw this amount of bubbles. And what is in those bubbles, Josh?
So that is carbon dioxide. That is the gas that those yeast excrete when they ferment those sugars that are within your flour.
Perfect. Okay, so now what we're going to do is we're going to show you guys how to feed our starters. So these are living organisms, and they do need new sources of food or they will die. So you're going to have at some point, a bubbly mixture. So the first step we do when we go ahead to start to feed this is you want to make sure you mix all of that back up. So you're just going to take your spoon and give it a nice, good mix. And that's going to release all of that carbon dioxide out of your starter, and it's going to kind of flatten your starter a little bit. The more mature your starter is going to be, it's going to get pretty stretchy. So it's not going to be really like liquid. It will be that really stretchy kind of consistency a little bit. So then what we're going to do is you want to save 30 grams, which is about 2 tablespoons of starter a little bit. What I like to do is I like to pour all of my starter out and then put 30 grams back in. You don't have to have 30 grams specifically. I tend to shoot between like 30 and 35-ish grams, which is 2.5 tablespoons if you don't have a kitchen scale. The really wonderful thing about this type of experiment is you don't have to be too terribly precise with it. You can have a little bit of leeway while you are making your starter, because it's a really happy system. It doesn't take a lot for it to do. So while I weigh this out, Josh, what happens if you change the flour that you have been using to a different type of flour?
So thankfully, starters are very adaptive. So if you change your flour, it'll probably get upset because you're changing what it's been eating for a while, but they do get adaptive, and so you'll start to see it behaving the same way, rising and bubbling. It's almost as if you stopped eating the food you regularly ate and you completely switched to dog food. You'd probably be upset for a while, but I'm sure at some point you'll be happy with it and you can still do everything that you like to do.
Perfect.
Hey, Josh and Mariah, we have another question.
Yeah.
What is converting the starch in the flour to a sugar that the yeast can consume?
Yeah, so that is the work of the bacteria that are in your sourdough starter. So the bacteria are very, very versatile, and they can eat a number of different starches in your flour. So what they do is they take in these very complex structured starches, and they will break them up and leave some sugar left, and that's what the yeast will eat. So it's a cooperative environment.
Great answer, Josh. So I have about 30 grams of my old starter left in my container. And now what we're going to do is we're going to feed it. We're going to give it some new food to munch on. It's just going to be like starting your new starter. So with your 30 grams of your old starter in your container, you're going to add 60 grams of flour and 60 grams of water. Again, you don't have to be 100% precise with this as long as you're making them equal weight. So that is about 60 grams of flour, and I'm going to add 60 grams of water. And then what you're going to do is you're just going to make sure that you mix it really, really well so that your flour, your water, and the old starter that you had in there becomes really, really incorporated. And you want it to be the same consistency that we talked about for when we just made our starter a couple minutes ago. Sometimes, like this one is coming out really, really dry, so I'm going to go ahead and add a little bit more water. Here, let me show you what dry looks like first, I guess, so you guys can get an idea of what that might look like. So this is like a really, really dry mixture. You can still see a lot of flour in there, and it's not really easy to mix. And that's what we mean by too dry. So I'm going to go ahead and add a little bit more water, and then go ahead and mix that up. And then that is how you feed your starter. Josh, how often are we supposed to be feeding it?
So for the first couple of days, your starter's alive, so probably 3 to 5 days. You don't want to feed it, or no, never mind. For the first 2 days, you want to feed your starter about once a day. So past that, you want to start feeding it twice a day. So probably whenever you wake up and then before you go to bed. Feeding it twice a day is how we're going to get that starter to be very vigorous by putting that pressure on it to perform and grow fast. Mariah, what do you do with the rest of your starter that you did not put in your jar?
Yep, so I have a bunch of starter left over that I put in a baggie, and you can do lots with this. You can bake with it. You can make sourdough pancakes, which is really, really yummy. You can make bread. You can make lots of different things with it. You can also give it to a friend, so then they now have a starter that they can do things with, or you can just throw it away. That does seem a little bit wasteful. However, these are all natural things, so you can either put it down the drain or just toss them in your trash. Either of those are totally good options. Perfect. Jennifer and Faith, do we have any questions?
So I do have a question for you. When you are ready to give your starter out to your friends, do you have to wait a certain time, or can I give my friend some of my starter tomorrow?
So that's a kind of like a preference. I like to prefer to make sure that it's a happy starter when I give it away, so then it can be ready for them to use right away. So I usually like to wait about a week before I start giving friends and family my extra starter. But that's a really great question. At any point you can give it away. You just have to give them some more instructions on how to feed it and how to keep it happy.
And for how many days do we need to feed it twice a day?
So you can do that until the end of time. So once your starter is vigorous enough for you to start baking, you can continue doing your twice a day feeding. But what you can also do is, if you want to take a break, you can put your starter in the fridge and you can start feeding it once a week. So that way you're not using that much flour all the time.
And then Josh, once you put it in the fridge and you want to make bread again, how are you able to then kind of warm your starter back up for it's ready to use?
So it's personal preference, but what I personally do is I take it out of the fridge probably before I go to bed, let it sit overnight, eat whatever flour it didn't digest, and then I feed it for about 1 day's worth, so 2 times, and then probably about then it's vigorous enough to use. It's important that you look at your height of your starter because that's the best indicator of whether it is ready to use.
So just for clarification, before you guys move on, we're not necessarily looking for an amount of time that we are feeding our starter before it's ready. We're looking for signs, correct?
Correct. Yep. And those signs are that it's really bubbly and rising up to be twice the height of when you start right after you feed it, and that it doesn't really have that smelly nature anymore. It does have, I mean, mine's not like the 1 month old one smells like sourdough bread. So that's okay, because that's its now happy smell. But we don't want to use it when it's really, really smelly. That's when it's kind of still pretty immature.
Mm-hmm. And the type of flour that we use there's no limitation, correct? We can use gluten-free flour, we can use whole wheat, we can use baking, all-purpose.
Yep, you can use whatever flour you want. If anything, whole grain flours are much better for starters. They're a lot happier with it because there's a lot more nutrition in there, and because they aren't bleached like a lot of all-purpose flours, there's more sleeping yeast and bacteria that are in that flour. So as you put more flour into your starter, you're actually bringing in more, and that can make your starter even more robust.
Yep, and especially in kind of the time we're living in now, stores are running out of certain types of flour. So if you go and you're looking for all-purpose flour or the type of flour you have been using, but they're out of that, but they do have almond flour or oat flour, that's totally fine. That actually happened to me with my really old starter. And you just have to give it a couple days for it to get used to it, and then it will start flourishing again. And then it's a happy, happy camper, and you can keep flipping back and forth if you need to. But sorry, go on.
Oh, no, sorry. Go ahead, finish your thought and then I'll ask.
I was just saying that, there's really no limitations. I just do want to say that each type of flour will act a little bit different. And that just means that it might take longer or shorter amount of time to get those bubbly signs that we're looking for to have like a really happy, vigorous starter.
It's also dependent on your location. Some of the yeast and bacteria that you get in your starter are actually from the air. So that's why the San Francisco sourdough is so famous, is because there are some yeast and bacteria that are endemic to just the San Francisco area that give it its own unique taste and behavior, such as the bacteria. I want to make sure I get this right. It's Lactobacillus san franciscensis. That's one, you don't have to know that, but it does vary a lot from where you are.
So we have another couple questions about, once your yeast is ready, how much of it do you actually need to put into making bread?
Oh, that's a really wonderful question. It's really dependent on the recipe you're using. Most of the recipes that I have seen use, it's like 120 grams, which is almost perfectly the amount of the discard that you're not using when you're feeding your starter. So what I have left behind from what I used today, that's about 120 grams. So I could actually use this to then go ahead and make a whole loaf of bread. But there's lots of different recipes online that use a little bit different amounts, and just go based on the recipe that you're using.
Yeah, so I want to make sure before we run out of time that we kind of go over some things you want to look out for in your starter. So there's a couple things you really need to pay attention to. So if you do see any drastic change in color in your starter, that means it is probably being populated by some bad bacteria or molds, and you want to throw it out. So if you start to see anything blue, orange, red, pink, anything that looks not like flour, that's probably a sign that it's been contaminated and you want to throw it away. Similarly, similarly, I was going to say, if you see anything fuzzy,
yep, that's what I was going to say too.
Thing.
Yeah, and that's one of the big reasons why you want to keep it covered, because we don't want any bugs or anything getting in there and trying to make it its habitat or anything like that. The other thing is, if you're past the smelly stage and you're into weeks 1 and 2 and it starts smelling again, that's also another indication that that might not, that something has gotten into your starter.
Yeah, but I think what's important to know is that if you do feed your starter on a regular basis, they are very strong, and it is actually that sour part of the sourdough that helps keep away a lot of those bad bacteria. So if you don't feed your starter for maybe a week, like I accidentally did, it can get contaminated. But if you feed it like you should, if you're a good sourdough starter parent, then you shouldn't have any problems.
Absolutely. So what we're going to do is we're actually going to transition back to faith, so we can move on to our next section. However, Josh and I are going to be answering any questions that you guys may have in the Q&A section. So if you guys do have any more questions, please keep asking. We will answer them in our Q&A box. So thank you guys so much for watching.
Thank you.
Thanks, Mariah and Josh, for taking us through how to make our starter and giving us some instructions on what to do after that's all done. We want to make sure that the next thing that we do is think about how yeast are these perfect organisms that we can learn from. I also want to take a second to introduce Jennifer Johnson, who's a Senior Research Associate at Berkeley Lab at the Joint Genome Institute. She's been answering your questions online with her scientist in training, Erin. So Erin, how are you doing today?
Good. I'm going to show them right.
Good. Did you finish your starter?
Yeah.
All right. So great, and thanks, Jennifer, for answering the questions so far. We're going to now transition back to Robert Riley, who's at the Joint Genome Institute. and he's going to tell us a little bit more about the science behind yeast and what we learned from them. So I'm going to start sharing my screen again so we can get some pictures up here. And it's over to you.
Okay, thanks, Faith. And so, yeah, I'll tell you some of about some of the cool things about yeast and some of the things that we've discovered from yeast genomes. And so first, what is a genome? What is DNA? If you got only one answer to that question, I think, to simplify a little bit, it's DNA is the basic building blocks of living things, or at least of our genomes. And so these are just some animations here showing you that it's made up of A's, C's, G's, and T's that are arranged into sort of a double helix structure when that comes back. Okay, so next slide, please. So it's the A's, G's, C's, and T's, and that makes up the DNA. So when we look at the DNA of lots of different organisms, we can start to see the relationship between those organisms, and we come up with a thing called a phylogenetic tree. And it sounds like a pretty complicated thing here, but it's pretty simple. Phylogenetic just means relating to the evolutionary development of a group of organisms. And a tree is just a diagram that we use to illustrate those phylogenetic relationships, just how organisms are related to each other through evolution. And we infer that from similarities in their DNA. So then if we look, next animation, please. If we look at this tree of life, this simplified tree of life, I've just circled the gram-positive bacteria and the fungi. That's where the bacteria are and the yeast are that are helping us make our sourdough. So although they look the same under a microscope, just these unicellular things, they're quite far apart in the tree of life, and at least a couple billion years of evolution separates them. And actually, we're in the animals, we're right up near the fungi, so we are much more closely related to yeasts than we are bacteria. So you can use phylogenetic trees to visualize not just the entire tree of life of everything that's alive, but you can zoom in on a group of organisms that you're interested in, for example, fruit trees. So here's a phylogenetic tree including some of the prunus species of plant, which includes apricots, plums, peach, cherry, almond, and they're also quite closely related to apples and pears. And so you can see how things you're used to encountering every day and eating are actually related phylogenetically. Often they're very closely related, and so closely related that members of the different species can breed together. So when Leo talks to us in a few minutes and walks us through his garden laboratory, he'll tell us a little bit more about that. So this is just a teaser for that. But you can really use the phylogenetic trees to look at any group of organisms and how they're related. And so this is a phylogenetic tree of some yeasts that we sequenced their genomes at the Joint Genome Institute. And we were mostly interested in their physiologic properties, such as their ability to turn sugar and starch into things we care about and can use, such as lactic acid and acetic acid that make our sourdough bread sour, and many other useful compounds such as biofuels and drugs and vitamins and flavor molecules. And then to get back to this, why do we even care about yeast or any, organism that we study in the lab? And it's a time to, it feels opportune to point out that the DNA building blocks and much of the genes that are in a yeast are very similar to some gene that's in a human being. So we share, we're made up of the same genetic stuff, and even, evolution doesn't always reinvent the wheel. It usually reuses an existing one. So same building blocks. The A's, G's, and T's are the same for humans and yeasts and just about everything else that's alive. So in biology, in scientific research, sometimes you go looking for one thing, but if you're keeping your eyes open, you might find something unexpected that is cool and maybe even more interesting than what you originally set out looking for. So this is a little story about that, about finding the unexpected within yeast genetic code. So what is a genetic code? So I told you what the DNA is, and this is kind of the fundamental process of, what does DNA actually do? What's it for? And just to give you one sort of simplified answer to that, the DNA is translated to mRNA, and it's just instructions for making a protein. So each codon, which is 3 nucleotides, specifies a particular amino acid that is a building block of the protein. And that's how all the proteins in cells are made, including the enzymes in yeast and bacteria that ferment, sugars into lactic acids and so forth. So all of this process is shared by all living things: bacteria, yeast, human, cats, apricot trees, everything. And it's all done according to this set of rules, which we call the genetic code. So it's just kind of can be visualized as this table of how the 3, any pair or any triplet of nucleotides specifies some amino acid. And it's rather remarkable that although evolution changes so many different things about an organism, it never changes the genetic code, except for on extremely rare occasions. And we knew when we sequenced these yeast genomes that one of the codons, CUG, was translated different from everything else. It was translated into the serine amino acid. So we knew about that. It was actually the only time this had ever been seen in the nuclear genome of a eukaryotic organism, that is a plant, animal, fungus, etc. And so we sequenced some more genomes and we analyzed their genomes and inferred a genetic code, and we found a different genetic code in one yeast for this same codon. But that was only the second time in that, a recoding of one codon to a different, from one amino acid to another, had ever been seen. So that was cool and rare and unexpected, and really quite just unusual, I should emphasize. So when we display these genetic code changes on the yeast phylogenetic tree, and each of these genetic codes is shown in a different color. So blue is the normal genetic code, and the pink and the yellow are the alternate genetic codes that are present in the yeast. We see they're sort of patchily distributed throughout the phylogenetic tree, along with evidence in their genomes that the yeasts were infected with some kind of pathogen, something like a virus or a killer plasmid. And viruses, when they infect an organism, they do so by exploiting the organisms, by having the same genetic code and using the organism's translation machinery to make, instead of the host's protein, make the virus's protein. So it's it's really quite evil from one point of view. And so this led us to conclude that that why did the yeast do this? That they altered their own genetic code in order to respond to an infectious pathogen. And I'll leave you with that thought. And just a few more things that yeast can do for us. So we discussed that the yeasts are useful in food such as our sourdough bread, and also brewing, beers. They're also useful in other ways. They can, through their fermentation abilities, they can produce ethanol, which is something that cars can potentially use as fuel. And their uses don't stop there. Through the modern genome editing tools, where we can, introduce genes into the yeast that are not naturally found in them, we can get yeast to produce all sorts of useful drugs and medicines and dietary supplements and fragrances and flavors. And so with that, if there are any questions, I can take them, and otherwise we will get back to the experiment. Thank you.
Thank you, Robert. I'll say again, how unique is it that yeast can change their genome, or they can combat these pathogens by changing their genome? Is that seen in any other organism?
So yes, in a way, in bacteria, it's well known that they change their genetic codes to fight viruses. But as far as I know, the only instances of that have been seen are ones where the code for an amino acid is changed to a stop codon, or where a stop codon is changed to amino acids. So those genetic code changes that happen between bacteria and the viruses that affect them usually involve the stop codon. So it's quite rare to see the kind of genetic code where a codon is reassigned from one amino acid to another. And this case in yeast is these two cases in yeast are the only ones that I'm aware of for a nuclear genome of a eukaryotic organism. So the bacteria and mitochondria are a little bit of a different story. But basically in all of eukaryotes, this is the only time that it's ever been seen that I know of. So really quite rare. And so it's not an easy thing for an organism to change its own genetic code, because you would have to, it's hard to explain here, but you would have to basically get rid of that codon from all of the genes that are in that organism that are being translated to proteins. Because if you change the genetic code, those genes would now be translated wrong, and they would be producing possibly non-functional proteins, and it would be very deleterious, a disadvantage to the organism. So it's changing an organism's own genetic code is not easily done, and that's why it's so rare.
Awesome. Thank you for that answer. We're going to switch over now back to Jennifer and Aaron, and they are going to introduce our last speaker for the day. Thanks, Robert.
Thanks, Robert. Thanks, Faith. So I'm really excited to introduce our next speaker. His name is Dr. Leo Baumgart, and he is one of my lab mates. Hi, Leo. He works at the JGI. He is plant genomics researcher, and he is going to tell us about his garden with his wife JC, who is also a scientist and works on yeast and engineering them to produce useful chemicals. You're still on mute.
All right, there we go.
Sorry.
Hello, can everyone hear me? I think so now. Anyway.
Yes, we can.
Yeah, we can hear you.
All right.
Okay, thanks for the introduction, Jen. So as Jen mentioned, I work at the Joint Genome Institute as well, and I study plant genomics there, which is basically trying to understand what are the effects of the different genes in a plant's genome, and how does that lead to different traits. So we talk about genotype and phenotype. The genotype is the combination of genes that is in the DNA in the genome, and the phenotype is the way it looks. So I'm actually messing around with that a little in my kind of at home in my yard, but first let me just take you through a quick tour, show you all the things I'm growing. I took out my winter vegetables recently, so everything is pretty small right now, but let's take a look at the different plants I have. So over here, start at the end, this is where I'm growing my beans. These are beans I grew last year, and I collected seeds from them and started these plants indoors under some lights. That's something you can do just under a light bulb if you want to get started when it's still cold. It's getting pretty warm now though, so they're pretty happy out here as long as they get enough water. And then here I'm growing some cucumbers, and these will probably fill in the space pretty well eventually. And the last thing I'm growing over in these planters, I have some peppers. They all look kind of the same, but when they grow up, you'll be able to tell the difference. I have several varieties of peppers that I also started from seeds. Cool. So now let me show you, I have another set of planters over here, and I'm going to show you, this is kind of more my experimental side. So over in the back here, this is some corn that I'm growing, and next to it, I'm growing a new variety of peas that I'd never tried before. And you can see they have this kind of cool mutation where they grow crazy amounts of tendrils like this. So they're really good at climbing up. And that's why I built this pyramid kind of for them so they can all climb up to the top. And then lastly in this planter here, I have some tomatillos, which are kind of similar to tomatoes, but they have these husks around them if you've ever eaten tomatillos, and they're mostly used for making sauces. Okay, so now this one here is kind of my test plot. These are all things I'm trying out, but over here, I'm growing a lot of tomatoes, and you can't really tell that well because they're all pretty small plants still, but each of these is actually a different variety of tomato. Some of them are just new ones I'm trying this year. Other ones are tomatoes that I crossed from tomatoes that I grew last year. So, for example, I had these two tomato varieties that I liked. One was called Cosmos, and they were pretty big tomatoes. One was called Blueberry, and they were these really small tomatoes. They were purple and had really good flavor, but they were pretty small. So I wanted to see if I could make a tomato that's a little bigger, but still has the really good flavor and the cool purple look of the blueberry tomato. So that's what I crossed the two parent plants together last year, and this is what came out when I grew the seeds. So I don't know yet what the tomatoes are going to look like. I actually have no idea, but I'm hoping since both the parents were pretty good, that this one will make good tomatoes too. And one thing you might notice, compared to the other plants in here, this one is actually quite a bit larger, quite a bit taller, more vigorous, and has more leaves on it. And it's kind of an interesting example of something that's called hybrid vigor. It's when you cross two different members of the species that have different genotypes, you cross them, and oftentimes the first offspring generation, it's called the F1 generation, shows this hybrid vigor. It grows extra well. And you can think of it as basically getting the best of both parents. It gets the best genes from both worlds. A more technical explanation, if you're interested, is that it's because the parents have some recessive traits which might not be that good for growth or vigor. And then if you get one copy from each parent, they can compensate for the recessive traits in either parent. So this F1 is going to be, it's not an inbred line yet, meaning if I get seeds from this, they might not look like the parent. If you really want to have a type of tomato that comes out the same every year, every time you pick seeds, then you have to cross it many, many times and always select for the one that looks like what you're trying to breed. So this is traditional breeding. It's a long process. It takes many, many years. And it's how we have all of the variety of crops that you might see in the grocery store at a farmer's market today, like these purple tomatoes. Some farmer a long time ago saw tomatoes that were a little bit purple and started crossing them over and over, and every year took the more purple ones that they liked. And eventually they got to this trait. So that's kind of what I'm doing just for fun as an experiment at home. I'm trying to see what is a new variety of tomato that I can come up with that no one's ever made before.
Someone's asking, how did you make the hybrid? Is this something that we could try at home?
Yeah, it's actually pretty easy. It was my first time trying it last year, and I just watched some YouTube videos. It's pretty simple. Basically, both parent plants have to be flowering at the same time. And then you take like a young flower from the female plant. You could choose either as the female. And before that flower opens, it hasn't made pollen yet. So you take off all the parts of the flower that would make pollen, and now it can't self-fertilize. It needs pollen from some other source. So now you go to a mature flower of the plant that you're going to use as the male, take some of the pollen off of it with a paintbrush with your finger, or just take the whole flower, and you brush it on, and then you mark it with a tag so you know if a tomato forms there, that's going to be the hybrid tomato, and the seeds from that tomato will give you the hybrid F1 plants. Cool. So like I told you, if you want to breed for traits, you have to kind of select them over many, many generations. And you have to also start out with a little bit of that trait. You can't come up with a purple tomato if you never find any tomatoes that are even the slightest bit purple. So what would you do if you wanted to introduce traits that weren't available in any member of the species that you're breeding? So Jason's going to tell you a little bit more about that. And as Robert mentioned earlier, she's going to be talking about stone fruit. All right.
Yeah, so luckily we have a great example of a bunch of different trees in our yard that are different species but are very closely related. And there's some really cool stuff that I want to show you. So I'm going to start by going over to our plum. All right, so this is our plum tree. And so I just want to point out a couple of traits about this plum tree to kind of contrast it with some of our other stone fruit. So plums, if you've ever had one before, these are not ripe yet, but they have a very smooth texture. And they have a really nice kind of tart flavor, but their flesh is pretty soft. If you've ever bitten into a plum, it just drips everywhere, and it's not a very fleshy kind of texture. And so now we're gonna I want to contrast that with an apricot that we have. So let's go over there. Okay, so here is our apricot tree. And so here, the fruits of an apricot are, if you've ever tried them before, very fuzzy. And the flesh of an apricot is a little bit more firm, right? You can kind of take out the pit and you have pieces of an apricot. And so, so this is where you can start to, because these plants are, a plum and a apricot are two different species. But as Robert showed earlier, they're pretty closely related in terms of their phylogeny. And so what a farmer did a long time ago was say, gee, well, I really like the smooth skin of a plum and I like the flavor of a plum, but I really want to keep that kind of fleshy texture that an apricot has. And in this instance, he wasn't able to probably find a plum that had nice firm texture, but he was able to find that you can cross an apricot with a plum because they are closely related enough. And so the outcome of that cross is a pluot. And so the pluot is over here. All right, so here's a pluot. And so this fruit, you can see, let me make sure you can see. So pluots have more of a smooth texture, like a plum, but when this becomes ripe and I can taste it, I'll find that it has that fleshy texture like an apricot. So here is an example of where the farmer was able to do a cross and get sort of the best of both worlds, select what he was looking for by getting a combination of the traits of both of the parents in the. And it's not just the fruit that get this combination, it's actually the whole plant that gets a combination of the traits of both of its parents. And so to kind of illustrate that in the leaves, I picked a plum leaf and an apricot leaf. And so you can kind of see how they differ. The apricot leaf has sort of a pointy tip and a very broad leaf, whereas a plum has sort of more rounded tip at the bottom. And if you look at the leaf of a pluot, which is a cross between the two, you can see it's kind of more narrow, like a plum leaf, but it has a pointy tip, like a very pointy tip, like an apricot. And so this is just another example of how you can cross, even though they're different species, two very closely related species, and you'll end up getting a mixture of the traits, and you can use that to select for a totally new plant that doesn't exist in nature. And we've done that a lot through farming and selective breeding. And so that's kind of the old-fashioned way of doing this. But now we have new technologies. We are starting to understand genomics and molecular biology to the point where we can actually make some of these changes in a much more direct way. So to give kind of an example of that, let's say I'm a farmer and I've got apple trees and plums on my field, and I know that the apple trees are super disease resistant. Unfortunately, there are far enough distinct species that I actually can't cross an apple and a plum and get sort of a disease-resistant plum. But if I know that gene, the technology is there to move that gene from the apple to the plum and be able to create a disease-resistant plum without having to go through many, many rounds of selective breeding. This is the kind of thing that scientists are working towards now. You know, it's very sophisticated. We can do it a lot in yeast, which which Robert talked about earlier, and Leo's working on making this technology possible for us to do in plants. And so with that, I'll pass it back over to Leo.
All right, so I hope we gave everyone an impression of kind of the lab we have at home for fun. We don't have a very high number of replicates, you know, N of 1, just because I don't have a whole test plot of planters. I wish I did, though. But yeah, I think it's like kind of a fun hobby. It's something that's easy to do, and you can really grow things without any sort of expensive lab equipment. And just by watching some YouTube videos and trying it yourself, you can grow your own tomatoes. And you can start crossing them and make a tomato no one has ever made before. So that's what I'm working on. Any other questions that we have?
I think you answered them, Leo. So mostly people were interested in how do you hybridize those plants, and you said you can find them on YouTube, correct? Or some videos that can help out?
Yeah, if you just look at crossing tomato plants, there's a lot of different resources, and it's quite simple. There's also some university resources that I've seen online with pictures, but I found the YouTube videos to be the best to kind of show you exactly what to do.
Awesome, thank you, and beautiful garden. Thank you for sharing with us.
Thank you.
So lastly, I'm just gonna share my screen one last time, everybody, and encourage you to, if you are using our methods or the methods that we showed you guys here to start a sourdough starter, please tag us and take pictures and share them. We're sharing a picture from Twitter from Marilyn Sargent, who's a multimedia producer here at Berkeley Lab, and she shared her beautiful sourdough bread that she baked with a starter that we did a few weeks ago in April, and it made us very hungry. So if you do start your yeast starter and make some bread, please share that with us. Also, if you want to know more about any of the laboratories that were talked about today and the institutions that were talked about today, here are some of the social media handles that you can follow us on and learn more about Berkeley Lab, as well as the Joint Genome Institute and the Joint BioEnergy Institute, which is JBay that we've been talking about. So thanks again for joining us, and please go to our Live Science Series site. We'll be doing more of these and talking with more of our scientists and connecting with some simple activities and hobbies that we do at home. So have a great weekend, everybody.